A couple of announcements about what's happening next week. On Monday, we'll have a video on smallpox and Marburg fever and cool viruses from hot springs. That'll be presented by one of my graduate students. There will be questions on that video come the exam because, I won't be here, but of course it's one of the lectures. I'm also going to post the poxvirus lecture from last year on YouTube. So that will be there as well, as mostly for background information. On Wednesday next week, we're going to have Dr. Luis Ruedas from the biology department. He's going to talk about virus ecology, but really specifically virus ecology of Hantan viruses. Best known of these are the sin nombre virus, which is another one of these hemorrhagic fever viruses, which is pretty prevalent in the rodent population here in Portland, which he and his lab found a couple of years back now. So that's what's happening next week. Again, the material in both of those lectures will be on the final. I don't know if I'm going to be able to post the video on YouTube, so I'll see how that works. I can make no guarantees as far as that's concerned, and I don't know about Dr. Ruedas, whether he'll have that available either. So, second thing, a number of people have already asked me about the exam. We're going to go over a couple of questions on the exam. You'll probably have noticed where the high score is. There are two scores which are way beyond the rest of the class. So those are clearly going to be cut off in terms of finally doing percentages, but it's hard to do that in D2L. So in terms of figuring out where you stand grade-wise, I would normalize to 75 total points at this point, rather than I think 85, which is the current high. So that'll give you a bit of an idea of where you stand. If you still have questions about that, let me know. I'm going to try and get D2L to talk to iClicker and have those posted by later today. Yeah? 75%? No, 75 total points. And so then you can calculate the percentage, what you have relative. So that'll be your denominator. Right, exactly. So not instead of 100, it's out of 75. Other questions about sort of grading, et cetera? When we're done with class, your Scantrons are up here, so you can pick them up if you like. Okay, that being said, there were a couple of questions on the exam that I did want to go over, and so those will be these here. These are ones that lots of people missed, which again I presume is because I didn't do a terribly good job of explaining the question, or the answer anyway in the class. So the translation of the longest polypeptide in the coronavirus genome is similar to translation of a bunch of different polyproteins, some of which aren't even really polyproteins because they aren't polyproteins. Poliovirus polyprotein and HIV-1 polyprotein are the only two here. And I posted the figures from the text and also from my lectures here. You think about how you get ORF1A and ORF1B fused to each other, which is the big polyprotein, the largest polyprotein in the coronaviruses. It's frame shifting that happens here. And of course, that's exactly what happens when you get the GAG-Pol polyprotein. So it's a frame shifting event which takes place. So I thought that was a little bit more straightforward, but I guess most people didn't. Second one is, what's the best evidence that the reservoir for SARS is bats instead of civet cats? And then various differences here relative to sequences. So the sequence of the bat virus is more similar to human SARS than the civet cat virus. Actually, the civet cat virus and the human virus are more similar to each other than the bat viruses are. More sequence diversity in bat coronaviruses than human coronaviruses. This is definitely true. But it says nothing about civet cats and or reservoirs. So there's more sequence diversity in civet cat coronaviruses than bat coronaviruses. As far as we know, there's only one civet cat coronavirus, so there can't be more diversity there. So basically, the only one you have left is there's more sequence diversity in bat coronaviruses than civet cat coronaviruses. And that gets back to this table here. And yes, there's much more sequence diversity in bat coronaviruses. which tells you it's quite possibly a reservoir, but that civet cats are a low diversity means it's probably highly unlikely to be a reservoir. Now, we also could have talked about phenotypes, what kind of disease or not, as the case may be. But this is, in fact, the first guess, of course, when people were looking at SARS, they thought it was a civet cat because the sequences were so similar to each other. But again, through more data and looking at the whole variety of coronaviruses that are present in bats, it's almost definite. Again, almost definite. This is biology, it's science. We're not going to know 100%. But it seems much more likely that the bats are the reservoir for this. And that brings me to another really quick aside of, I don't know how many of you, are looking at MERS. Again, there's been a couple of MERS cases now in the US, Middle East Respiratory Syndrome virus. I was actually interviewed for being an expert on MERS, which I thought was a little odd, but ended up on the web, and so there's a couple of postings. I'm now a local MERS expert. But the basic message is 500 times more people die per year from flu than have ever died from MERS. So just perspective-wise. And at least that got quoted. The other thing that I said didn't get quoted. But the answer, my question is, are there any foods you can eat to boost your immune system? And my answer was, there is no such thing. And unfortunately, that didn't get quoted. But so be it. The next, in fact, in the web posting, it actually says, eat these, immune system boosting foods. Say, the media. We may get back to that a little bit later on today, too, so we shall see. The last question that I wanted to go over just briefly here has to do with the overlapping terminators in the SV40 genome are probably important for transcriptional silencing, translational replication, anti-splicing activity, post-transcriptional silencing, or all of the above. And what I'm guessing here is that RNAi, everyone said it was RNAi. What is RNAi? RNAi is post-transcriptional silencing because you have double-stranded RNA that forms here with these overlapping transcripts. And so when you've got double-stranded RNA, that leads to post-transcriptional silencing by chewing up the messenger RNA through Argonaut and the whole RNAi machinery. So those are the only ones that I wanted to go over in a little bit more detail. Are there any other questions on the exam at this point? Also, feel free to email me if you have questions about specific things. Okay, so let's move on, getting bigger and bigger and bigger now as far as the double-stranded DNA viruses are concerned. Pox viruses on Monday are not quite the largest, but the largest of the animal viruses that have double-stranded DNA genomes. But this is sort of continuing on from what we were talking about before the midterm, the papilloma and polyoma viruses. So adenovirus, sort of the classic icosahedral virus structure, linear, double-stranded DNA virus. It's basically bigger and more complicated than these other ones. But relatively speaking, in terms of how it's replicating, and particularly in terms of what all of the research has been done, it's pretty similar to the polyoma viruses, at least in terms of host interactions. In terms of the virion structure, this, of course, is really quite different. And so we'll talk quite a bit about that. A couple of concepts here. Difference imaging, which is unfortunately now getting a little bit out of date, but the structure of these virions is really pretty amazing, and the, I think, largest structure for which we have atomic resolution structures is now these adenoviruses, or different forms thereof. We haven't talked that much about alternative splicing, mostly in terms of the retroviruses so far, but these adenoviruses are sort of the kings of alternative splicing. And in fact, where splicing was originally discovered was in looking at the late genes of adenovirus. Same thing is true for formation of PolyA tails. So this whole really complicated structure comes from alternative splicing and alternative tailing. cell cycle regulation turns out to be extremely similar to the polyoma and papillomaviruses. And so we'll talk about how that's working. The replication of the genome is quite different because it's a linear DNA as opposed to a circular DNA virus. So there's one of the big differences that we have from the papilloma and the polyoma viruses. The way that that works is through the presence of a terminal protein, which should sound a little bit familiar relative to the polioviruses, the picoRNA viruses. There also are inverted terminal repeat structures at the end of the genome, and that has to do with how the replication is taking place. So again, we'll talk about a little bit where adenoviruses come from, a little bit about adenovirus disease. It turns out adenovirus disease is not a big deal. One of the things that I did want to mention here in this micrograph, these icosahedral particles are the adenovirus, the large ones. We also see the arrowhead up at the top there. That's what we're going to be talking about next Friday. Those are the adeno-associated viruses. These are the really small single-stranded DNA viruses which often come along for the ride with these adenoviruses. The structure, we'll spend quite a long time talking about the structure because it's so gorgeous, but also because that really allows us to understand a lot about how these viruses are being put together and just some real fundamentals about how proteins fit together in terms of structure. A little bit about binding and entry. The binding and entry is pretty straightforward. And then transcription and replication being the really important differences here relative to the other viruses, and release, which is pretty straightforward. Where do adenoviruses come from? Respiratory and eye diseases. That's where most of the adenoviruses that people study come from. There are a whole bunch of them, 50 different serotypes. So if somebody talks about adenovirus type 2, type 5, type 50, etc. Actually, the last I heard, they're up to 55 serotypes. These are all different kinds of adenoviruses. There's a huge amount of diversity in these different adenoviruses based on serotypes. So people know what serotypes mean? So it's different antibodies. So antibodies to one virus will not necessarily cross-react with antibodies to another virus. So it tells you something about the surface structure of these different viruses. Backing up a little bit, they were originally found in adenoid tissue. People thought they were the common cold virus, but it's not true. Main reason that we're interested in adenoviruses is that they're wonderful tools for understanding, and I should have structure up here as well as splicing, general studies of transcription. And if you look at some of the older papers on transcription, they all talk about now how transcription works. Well, 90% of probably the first 4 or 5 years of transcriptional regulation in eukaryotes. Transcriptional regulation in eukaryotes was what's happening at the major late promoter or any of the other promoters in adenovirus. So really very much a big study of that. More recently, adenoviruses are being used for gene therapy purposes, and there's a lot of that going on right now. Probably 10 to 20% of current gene therapy trials are using adenoviruses, partly because these are tumor viruses, but tumor viruses are a lot like the polyoma viruses. They're only tumor viruses if you really push them really hard. Generally, they're not going to be causing any kind of tumor. We looked at the structure of these viruses before. This is from a slightly older cryo-EM image. They've got 11 different proteins in the capsid structure, let alone what's present on the inside. So these are very complicated capsids, but still very symmetrical relative to each other. They've got a T equals 25 symmetry, but it's a pseudosymmetry again because you've got lots of different proteins that are there. The main protein that's present there is the hexon protein, and this is the high-resolution structure of just individual hexon proteins. So if you crystallize hexon proteins just by themselves, this is the structure that you get. Of course, you also have differences of the five-fold axis of symmetry, where all these spikes are coming out. That's nice because it tells you where the five-fold axis of symmetry is, makes it easy to find. But also, these are very different proteins. So you've got 12 of these, again, regular icosahedra. The penton is interesting because it's got not only a structure which forms at the base of the penton, so at the literal five-fold axis on the surface, but also then these fibers and knobs that come off of that. And so that's the classic adenovirus, again, with these projections sticking off at the five-fold axis of symmetry. Until recently, people didn't really know where a lot of the extra proteins were in terms of the structure. So we've just talked about 3 here: hexon protein, penton base, fiber, and knob. Sorry, 4 proteins. But there are 11 proteins that are present in purified capsids. And so the question is, where are some of those other proteins? And this brings me to a really interesting technique that people used to use to get structures if you couldn't get high-resolution crystal structures for these viruses. So basically, you combined the cryo-electron microscopy, as you see at the top here, with the X-ray crystallography down here. So basically what you do is you take 240 of these structures and put them into that structure, and then subtract these from that structure and see what you have left. And that's shown graphically right here. If you take these hexon structures, so here's a hexon structure right here. You put 9 of those together, which is what you would expect in terms of electron density, and then you subtract these from the real structure that you get up the top here, and you end up with this electron density. And so this tells you where some of those individual proteins are. Now, this was great, very useful, until a couple of years ago when we actually got some crystal structures of the whole adenovirus. And this may or may not be in the notes that I posted yesterday, but I'll make sure that it does get there. So this was published in Science, again, a couple of years ago now. Each of the hexon proteins here in green, orange, blue, that's a green, yellow, orange, blue, all identical to each other. But the great thing that you can see here are also these extra proteins, the dark blue here and purple there. Also, you can see at the 5-fold axis of symmetry these penton bases and the penton fibers that are associated with them. At the same time, published, in fact, in the same issue of Science in 2010, this was the X-ray crystal structure, and again, probably the largest X-ray crystal structure of anything that we had to date. There was also a cryo-electron micrograph structure that was published. This particular one took, I think, 33,000 pictures of each of those original adenoviruses and then averaged them all together. And this is what they got. What's really amazing is that this structure from the cryo-electron microscopy is actually higher resolution than what they got in their X-ray crystals. So, and this is an example of that resolution down here. The wire model around the outside, this gray stuff that's a little bit hard to see, that's the data. And then where each of these individual amino acids fit into it, that's the model, and it's the model which is being projected here. But it's how well the model fits to each of these amino acids tells you the resolution of your actual structure. And it's really pretty amazing. You see some of these things like arginine side chains that have really nice density, these tyrosine side chains as well that are giving you really nice density. And that brings me to the movie of this, which, again, I've linked in the PowerPoints, but we can quickly escape out of here and take a look at this. So this is that whole structure from start. From the cryo-electron microscopy, basically spinning around one of these 5-fold axes of symmetry. There's a 5-fold axis right there. Here's that next 5-fold axis coming through here. But you can see the hexons quite nicely separated from each other. And then all of these accessory proteins that are present around the outside of each of those. And another way to look at this is to now take one of those faces. There are a whole bunch of movies that are associated in here. But if you just look at one of the faces, here, one of those triangular faces, you can see nicely it's T equals 5 icosahedron because you've got these 5 across here. Let's say T equals 25. And then here are all of those extra proteins, all the blue ones here, the red ones here. So you can really nicely localize exactly where all of these extra proteins are relative to the rest of the structure. Adenovirus is so gorgeous that it's also used as art. This is at the Cold Spring Harbor Labs, where they did a lot of work on first splicing and also transcriptional regulation. Curiously enough, this is on top of the wastewater treatment plant at Cold Spring Harbor Labs, where they have this. They also have a clock tower, but on their clock tower, instead of north, south, east, and west, guess what they've got? C, A, T, and G on their clock tower. It's really pretty amazing place. For a long time, Jim Watson was the head of the lab there. So this is the cartoon. Makes it a little bit easier to follow where some of these things are. We just take a quick look at the hexons. These are all of these gray trapezoids here. Then you've got our pentons, penton base, penton fiber, and penton knob. All of these things have numbers, which is ridiculously confusing. I don't try and follow any of them. You'll notice why I'm not calling it gene 2, gene 3, gene 3A, gene 8, gene 9, etc. Pentons, hexons, that's all that I can remember. But a lot of these structures, for instance, this one right here, again, it's 3A, but that's not that important. But people just drew these in cartoons, sort of guessing where they were. And now with those high-resolution structures that I just showed you, they know exactly where they are. And so you can design experiments to look at, well, okay, what is the role of that in terms of dissociation of the particle or interaction with the receptors and how all of those things are going on there. There are a couple of things that I did want to mention here. Of course, we've got our red circle here. That's around the terminal protein. This is what's attached to the end of your double-stranded DNA genome. The packaging of this genome, it's packaged in things that look like nucleosomes if you just look at the DNA, but the protein itself is really very, very different in terms of the structure. So it's packaged inside this particle. If you just look at the genome, what does it look like? Again, double-stranded DNA. Depending on which one of the serotypes you're looking at, it's anywhere from 30 to 36,000 base pairs in length. And again, it has a terminal protein bound at the 5' end. Double-stranded DNA, you've got two 5' ends. So each of the 5' ends has this terminal protein bound to it. And then there are these inverted terminal repeat structures at either end of the genome. And what that means is that if you read from 5' to 3' at one end, it should be giving you the same sequence, 5' to 3' at the other end of the genome. So if you start over there, and you'll notice I had to add a T because there's a typo in the figure in the textbook. First, it's like, what the heck is going on here? So CAT, CAT, CA over here, 5' to 3', CAT, CAT, CA. So that's the inverted repeat, and it's 100 nucleotides. So this is just 7 nucleotides here. But it goes for 100 nucleotides at either end of the genome. So again, this is where that mistake was. Needs to be changed. I told them about it in the first edition of the textbook. They still haven't changed it in the second edition. So that's what we have at the end of the genome. How do you get inside the cell? This is our figure from way back when we talked about entry. Adenovirus is C right here in the middle. So first you have interactions, not surprisingly, with the penton fiber and the knob at the end. And then the whole virion gets brought inside the cell, receptor-mediated endocytosis, and then the virion itself is transported to the nucleus. And again, double-stranded DNA, where you're going to have to replicate, all the replication machinery is going to be going on in the nucleus. So what's the binding that takes place? It's that knob structure again at the penton. It binds to the Coxsackie and adenovirus receptor. So adenovirus receptor, again, this was found because people were looking for what adenovirus actually bound to. Still not completely clear exactly what it's doing when it's not binding to virus. Interestingly, these adenoviruses not only have a primary receptor, but also a secondary receptor. So it's not just what's out at the end of the penton, it's also the penton base that interacts with integrins. And that integrin interaction is probably critical for getting it into the endosome. Once in the endosome, there's activation of a viral protease, and that viral protease, again, is how it's activated, change of pH that you have in the endosome, and then from there it breaks off basically the penton fibers. Once you get to the nuclear membrane, there's another protease which breaks down the rest of the structure in order to release the DNA. And so it's what these proteases are doing, what are their substrates that we're learning a whole bunch more about now that we have a higher resolution structure. We can look at where each of these individual proteins are, where they're getting degraded. Another way of looking at adenovirus binding and entry is here. It's just a link down here at the bottom. You have the knob at the end of the penton fiber interacting with the receptor, but then also co-receptors interacting at the penton base structure. These are usually the integrins, and again, those are the ones that are most important for getting endocytosis to take place in these particular genomes. So, endocytosis, proteolysis takes place. You cleave off these guys. That allows the virus to escape the endosome. Quite how that happens is not completely clear. That virus virion particle minus the penton fibers gets transported to the nucleus. The nucleus, you then release the genome. Now what happens? Well, now, of course, you need to make all of the genes that are present in this genome. And this is the map of adenovirus type 2, best studied of all of these. It looks like a god-awful mess, to be perfectly honest, when you first start looking at it. There are 8 promoters that are present in the genome, but they're also split up, just like we've talked about for many of the other viruses before. We've got the early promoters, intermediate promoters, and late promoters. And as far as we're concerned, it's mostly about the early promoters and the late promoters. So if we first look at the early promoters, there's E1A and E1B. These are the proteins that are most important for regulating what happens with the host. So again, as usual for these, what happens with early proteins, these are ones that are going to be important for regulating host functions. Then we have the E2s, E3s, and E4s. And again, they're all important for regulating host functions to start with, E1A and E1B. And then they're proteins which are important for replicating your genome. So we'll see this one's a DNA binding protein. This one's a DNA polymerase. So first, regulation of hosts. Second, regulation of actually making the proteins that you need to replicate your genome. Then you have these intermediate RNAs. It's not entirely clear what those are doing. And then what do you need to make virus particles? It's all these late proteins. Remember, there are 11 of them that are packaged in the regular adenovirus genome. They're all made from one promoter, and that's why this was such a good system to study. It's one promoter. It's a really, really good promoter. It makes tons of RNA, tons of different RNAs that all have the same leader sequence at one end, but then multiple different 3' ends. And those multiple different 3' ends are generated both by alternative splicing, but also by alternative tailing. And we'll talk about the alternative tailing a little bit later on. There's one other thing that I wanted to mention here. That's these guys, the viral associated RNAs. These are RNAs which are expressed, and they're functional RNAs. So they're not encoding any kind of protein. They seem to be important for regulating PKR that we talked about before, protein kinase R, which is leading to the interferon response and then blocking translational initiation. So PKR is a cellular defense mechanism. These viruses deal with PKR by making these specific viral RNAs. So I mentioned splicing. This is probably the most famous splicing image that anyone's really ever looked at. This is from a paper by Phil Sharp and co-workers. This is looking at part of the adenovirus genome, about half of it, where they take the messenger RNA for the hexon protein. So again, that's the one that there are 240 copies of in the viral capsids, what you need the most of any of these proteins. So you need lots and lots of that RNA, relatively easy to purify. So if you take this RNA and you hybridize it to DNA from, again, about half of the adenovirus genome, what you see is that that RNA hybridizes with the DNA through the whole coding sequence of that messenger RNA. But then the really bizarre thing is at the 5' end, it doesn't match up with the DNA that's right next to it. It matches up with DNA which is much further away. And so that's this messenger RNA that's here that hybridizes to the green part and then hybridizes to these 3 black pieces over here, giving you the introns. And so this was the very first example of introns that anyone had ever seen. At first they thought it was just viruses. Who cares? Turns out that, of course, most of our genes are being spliced in very similar ways. And this, anybody's in the molecular genetics class, I know a few of you are, it's black and white image, of course, and then the interpretation of these. They probably took literally hundreds of micrographs to get one that looked this pretty. That being said, both Phil Sharp and Rich Roberts ended up getting the Nobel Prize for this discovery of splicing, and then later on looking at that. So yes, splicing happens. It's really nice to study this in a viral system when you've got lots of RNA of one particular type. You can also get lots of DNA relatively easily. And so this allowed the discovery of splicing in the first place. So yes, it's happening in these viruses. What happens with splicing? Turns out that almost all of the genes in the adenovirus genome are differentially spliced, just like our genes. Almost all of them are differentially spliced. Let's start out with the first one of these, the E1A protein. Differentially spliced, 2 different RNAs, which are really not that critical. Most important is what these guys are interacting with. I like to think of the E1A protein as being really similar to large T antigen in polyomaviruses in terms of all the different cellular proteins that it's interacting with. Interacts with the TATA binding protein, so that's important for transcriptional activation. Interacts with a number of coactivators. These are things that are important for regulation of chromatin structure, allowing transcription to take place, and then our friend PRB as well. So PRB interactions with E1A, again, are extremely similar to those interactions that we saw with some of the small DNA viruses. Which one is this most similar to? Papillomavirus, which one? E6. E6 or E7? Take votes? I didn't ask you for clickers. E6, E7, E6, E7, E6, E7, E6, E7? It's E7. Yeah, so it's most similar to E7. But again, basically doing exactly the same thing. Interacting with RB, pulling RB away from the E2F. And E2F, I think I may have misspoken when I talked about this before. E2F is one of the proteins that is a transcriptional activator for adenovirus E2 proteins. So the interaction of E1A with RB allows the expression of E2. So just this whole cascade of processes. There's also these histone deacetylases. Again, these are transcriptional coactivators that are also interacting with E1A. E1A also regulates P53, but it regulates P53 in a really indirect fashion. So E1A, as I mentioned before, is a transcriptional activator. It activates this protein P14. P14 interacts with MDM. MDM is usually most important for regulating P53 structures. So if you have MDM2 around, it binds to P53 and causes P53 to be degraded. Turns out what it does is it causes the transport of P53 out of the nucleus, which is where it gets broken down. If you have P14 around, it binds to MDM. So it takes MDM2 out of the picture. So MDM2 can't interact with P53. If it can't interact with P53, then P53 is stable and still active. So you've got stimulation of the cell cycle with E1A leading to activity of these E2Fs, but also blocking inactivation of P53. Well, this could actually be a problem because usually P53 is involved in blocking the cell cycle. It's the guardian of the genome, right? So if it's the guardian of the genome, it's going to be blocking the cell cycle if you've got something nasty around like an adenovirus trying to make more adenovirus. So there's the other E1 protein present in adenoviruses, which suppress apoptosis and then also lead to degradation of P53, but in a later stage. So E1b, there are two different splicing products of E1b. Both of those suppress apoptosis, but also turn on P53 turnover. So those are the regulatory proteins as far as this part of the cell cycle is concerned. And again, it's just like what happens in the polyoma and the papillomaviruses. These are double-stranded DNA viruses. They need to replicate with double-stranded DNA. How do you do that? Particularly in differentiated cells, you've got to be stimulating the cell cycle in some way. So, yeah?
Sorry if we're already supposed to know this, but what's P53 turnover mean?
Oh, it's just P53 turnover is just degradation. So, you know, it's that degradation process of P53. Any other questions on this? So, what does adenovirus need in terms of replication? Double-stranded DNA, terminal protein at each of the 5' ends. You need to replicate this. This is not like a cellular DNA. It's not a nice double-stranded circle associated with nucleosomes with a nice origin of replication. How do you replicate this double-stranded DNA? Well, it happens really quite similarly to what happens in terms of things like picoRNA viruses, where they've got a terminal protein bound to one end. So here you have a terminal protein. This is the standard genome that you start with up on the upper left here. Then you have a terminal protein, which is made by some of those E2 genes. That then serves as a primer. Now, it's slightly different than what happens in VPG in the polioviruses. Here it's a serine that provides the OH instead of the tyrosine that provides the OH. But again, it's still an OH that's on the side chain of a protein. That OH pretends to be a 3' OH. Then the DNA polymerase can bind to the template, pull the two strands apart. And the pulling of the two strands apart is interesting. There's not a real specific DNA helicase that's present in adenoviruses. There's just a really good single-stranded DNA binding protein. And so that single-stranded DNA binding protein basically serves as the helicase. So DNA, just at 37 degrees, is going to be breathing a little bit. The two strands come apart relative to each other. You've got a really strong single-stranded DNA binding protein. As soon as those two strands come apart, it's going to bind to and hold that single strand apart. And so that's basically providing your helicase activity. Yeah? Is
the single-stranded DNA binding protein provided by the virus or is it by cell?
It's the viral single-stranded DNA binding protein. So it's one of those E2 proteins which is being made.
Okay.
Yeah. So again, it's and it binds much more strongly. So the single-stranded DNA binding protein, RPA, in eukaryotic cells doesn't bind anywhere near as high affinity as this does. So this binds. You end up with one copy of your genome. That's all well and good, but how do you get a double-stranded piece? Well, it turns out that this now depends on those inverted terminal repeats that you have at either end of the genome. Remember, it's 100 base pairs that is identical on one strand relative to the other. What happens is these single-stranded pieces can now make the frying pan structure, is highly technical term here, frying pan structure, where now you end up with a double-stranded piece of DNA just at one of the ends. Now the terminal protein and the DNA polymerase can bind there and extend their way through the genome, as well as replicating from this end of the genome. But this process, where you can have single strands that serve as templates, you can now amplify much more than the 1 to 2 to 4 to 8, you can amplify in a much more rapid process. And so this frying pan structure allows you to get many, many copies of the genome. Again, because it turns out adenovirus, and I don't have a figure of it here, but if you look at cells that are infected with adenovirus, they're just packed with virions. So you get really huge numbers of virions that are present in there. So this is how you get replication taking place. It's all dependent on your single-stranded DNA binding protein and then the terminal protein, which provides the primer for replication. In terms of transcriptional regulation, it's really all about the splicing, but not just the splicing, also the tailing, where you put a PolyA tail onto your transcript. So the major late promoter up here at the top, where you start, you get your primary transcript. Your primary transcript is pretty big, basically encodes all of those late genes, but those are not going to be what gets translated. What gets translated are one of a set of 5 different genes, or I should say gene families, all dependent on where you have the cleavage of this primary transcript in the PolyA tail, which gets added. So if you have, for instance, cleavage right after the late 1 segments of genes, then you have this piece with tripartite leader. All of them have the tripartite leader sequence. And then each of these individual ones can be spliced in different places at the 3' end. But it's this cleavage at the PolyA addition sites which leads to each of the individual fragments, and then splicing happens after that. So it's the cleavage first, splicing later, and each of these splice acceptor sites are going to lead to any one of these different classes. So again, we've got 5 different classes of these late genes, all of which are getting spliced. Each of them has this tripartite leader sequence, which is probably why Roberts and Sharp were able to find it, because every single one of these guys has this. Probably important for getting these messenger RNAs out of the nucleus, but even more importantly for getting good translation of each of these messenger RNAs. So it's the untranslated region here, which is really important. So you don't have the same N-termini on all of these proteins. You just have the untranslated region, which is just the same. As I mentioned before, you get a whole bunch of these virions in any given infected cell. You also now express some of the E3 proteins. Very confusing that E3 is in E should be early. Turns out it just has to be accumulated to relatively high levels. And once you have enough of this E3 protein, it leads to the death of the cell. And that seems to be apoptosis, basically stimulation of apoptosis that takes place. E4 is a protein that people have gotten really interested in much more recently, because E4 is another killer protein, but it works specifically in terms of cells that don't have p53 in them. So you remember most cancer cells don't have active p53 in them. You don't have active p53, you don't go through normal apoptosis, you don't block the cell cycle. E4 specifically kills these cells that don't have p53. In adenovirus infected cells, E1b has shut down p53, and so that process. But now if you can just express the E4 protein in a virus, that should just be causing death in cells that are lacking p53, which are then going to be your cancer cells. Yes?
So E3 is classified as a late because it takes a while enough to accumulate, but so does transcription start early?
Yes, the transcription is actually much earlier, and so all of the black ones here, and this is why I put this back up, these are all being expressed early on in terms of virus replication. And it seems to be accumulation of that protein. Takes a while to get enough of that protein. So that's not dissimilar to some of the lysis proteins that we've talked about, some of the bacteriophage. You just need to build up enough of this protein in order to have its activity. Okay, so that leads nicely to what people are trying to do with using these viruses. And again, I just mentioned this E4 activity. Tumor therapy, the idea is you make a recombinant adenovirus that has no E1B. So again, normally E1B is going to be leading to degradation of P53. You now have an adenovirus that doesn't have E1B. So normally it's not going to be able to replicate well. However, if you can get this virus to infect cells that don't have P53 in them, i.e. cancer cells, now this virus, at least in theory, is going to be able to kill off those cancer cells. And so I looked at this a couple of years ago. Everybody was really excited about this. Is going to be a great way to kill off cancer cells and oncolytic viruses. As far as I know, it was discontinued in 2003. So, but people are talking about now trying to do some of these things as well. There's also some people who are trying to use this for vaccine production. And the idea is here that you make a virus that's missing E1A and E2A, so it's not regulating the cell cycle, but it can still bind to and inject its genome into cells. Turns out that the CAR and integrin co-receptors are present in lots of different cell types, so these have pretty broad host ranges. Then you could have proteins that are important for vaccine production that you express on these. The last one of these viruses I wanted to talk about is the fat virus. I don't know how many of you heard about this, mostly a couple of years ago now. So the epidemic of obesity, so huge amounts of obesity are happening, particularly in the developed world, but also in the developing world. So what could that be? People said, okay, it's an epidemic, so therefore we have to have a virus that's associated with it, right? It's epidemic spread, obviously. Has nothing to do with lifestyle or anything else. So, or genetics or you name it. So this was shown, I think the first paper in 2000. If you infect mice with adenovirus 36, they do have an increase in adipose tissue, which is fat tissue. If you look at humans, about 30% of humans have antibodies to adenovirus 36 if they're obese. Non-obese is 11%. So there's an association with obese people and adenovirus 36. If you look at stem cells, they're stimulated by adenovirus 36, these adipose stem cells. The rat cells are not stimulated by these other adenoviruses, so maybe it's an adenovirus 36. And now let's click on this link. Oh yes, they'll look at the source here as well. So, obesity virus spreads like common cold. So, can be caught. Highly infectious virus which causes sniffles and sore throats. Nicoldurandar infects the lungs, then whisks around the body. Makes exactly so. Third of obese people had the rare and highly contagious virus. It's just 11% of thinner people. Okay, so they're associated with this. Yeah, people could be fat for reasons other than viral infection, so it's pointless for fat people to try and avoid infection. And the Pennington Medical Research Center, which is, interestingly enough, the place where all of this research has been done. Almost all of it, not completely. So, again, you want to be careful about these things. Yeah?
Could it also be susceptibility of obese people is the reason why they have higher antibody output?
So that actually brings me to the next slide. Thank you for the lead in here. So there's been quite a lot of discussion about this. Again, not surprising. This is all over the web and news and everything else. This Week in Virology discusses that, 18 and 101, for those of you who are interested in This Week in Virology. Particularly, number 101 discusses exactly that issue, is that there is a clear correlation between obesity and infection. And all of these cases were very specifically looking for adenovirus 36. They haven't looked for other viruses, general diseases, what those antibody counts are all about, etc. So basically the sort of take-home message here is there's certainly seems to be a correlation between presence of adenovirus 36 antibodies and obesity. But correlation is not causality. And so that, I think, is really the big take-home message from all of these papers, including this one. 78% of 19 children are adenovirus 36 positive and obese. Well, this is 15 kids versus 4 kids? And you're calling this statistically significant? Okay, if you just calculate your p numbers, that's true. But yeah, and so there does seem to be a correlation. But again, correlation and causality are two very big and open questions. Yes?
How significantly spread is it? Are there people that live close together anyway?
Oh, the spread of adenovirus 36, I think nobody's really looked at that. So far, it's been always antibodies. And then, whether it's in families. And one of the things they also talk about here in TWIV 101 is what you really need to do is a longitudinal study where you look at infection processes and what's happened over time. One of the really nice things about this, and experiments which could be done and may have been done and haven't been published, is if you go back and look at old serum samples, because they should still have those antibodies to these various different viruses in them. And so if it's really an epidemic that's happening just now, we should see a big peak in what's coming up in terms of these adenovirus 36. And I think the fact that not much has been published on this in the last 4 years probably gives you a pretty good indication about how far along this has actually come. So, but it's an interesting question. But again, it's a correlation rather than a causality that's happening right now. So with that, we basically talked about this already. It's Friday. Have a good week. Next week, I'll be back next Friday.